Good morning. Thank you very much for invitation. It's a great pleasure to be here to share what we are doing in University of Arkansas for medical science. At the beginning, I'd like to say we're at very preliminary stage, but we think it's a great potential what we are developing. I share with main concept, platform, preliminary results, and I hope on also on count on possible collaboration. It's called in vivo flow cytometry. It's very known. It's very known technique. Lots of application in vitro. Equation for cell analysis, for diagnosis, for basic sciences, to see apoptosis, cells property, etc. But the question is, why in vivo? Main idea is here. We call it in vivo blood test. Instead, conventional. Okay, how it works? Okay, we call it in vivo blood test. Instead, monitoring in ex vivo blood cell test, just few milliliters, we proposed many years ago in vivo blood test using different technologies. It's integration, nanotechnology. It's photoacoustics, flow cytometry, new nanoparticles. In a new method, it's, I cannot say sampling. We don't need sampling of blood. We try to monitor blood in vivo, naturally circulated in superficial blood vessels. We suggested several approaches with broad application. Today, I will focus on few of them. Just again, why in vivo? Because conventional approaches, it's just few milliliters of blood. For rare diseases or for rare abnormal cells, sickle, circulated tumor cells, bacteria, virus, you can miss if very small blood volume. That's why it's 1 cells per 1 milliliters. We have 5 liters approximately. That's why we miss 5,000 cells if we use sampling very small volume. So what does it mean if you miss 5,000? It's too late to treat cancer. Metastasis already developed. It's too late to treat sepsis because it's already developed. It's so on. That's why much as its principal new, say, diagnosis, early super sensitive diagnosis, great potential, but we are at very beginning. If we proposed potential impact, it's 1,000-fold increased sensitivity compared to conventional blood test. And we already developed preliminary prototype, clinical prototype, of course, animal models. We have several IRB protocols. We go to clinical trials. And we focus, we have a small group on 3 or 4 diseases. At the beginning, it was cancer, to detect early cancer metastasis. Sickle diseases we focus, infections, staphylococcus, and cardiovascular diseases. We go clinical trials, first melanoma breast cancer next year, we hope, if everything okay with IRB, necessary approval. But we mistakenly, during some testing, occasionally discovered another method. We can detect circulating clots. And it's a lot of complications related with clotting, thrombosis. I'm not going to cover all medical field. I just give some examples. Strokes. What does it mean? If it's clot or thrombus, it's aggregated red blood cells, platelets, different regions goes to brain. They block vessels, prevent blood supplies to brain. Stroke has happened. That's why what a question is a lot of factors: viscosity, blood pressure, stress leads to this disease. But definitely, final lethal events, it's clotting, blocking vessels. That's why it's last stage. If we focus, could we prevent stroke if we could detect early clots, clotting, etc. At least we can determine risk after that, or maybe to detect in time large dangerous clots in vivo using our technology. Equation: how many clots, what size can detect, and what's relation between clotting and final events. Nobody knows because no clinical device on market right now to find relations between final event stroke and clotting. What size, how many clots create this huge problem, huge market. Previous speaker told briefly about it, about this pricing. How it works? It's combination, just few scientific details. It's combination laser and ultrasound. It's photoacoustic effect, transformation, light and sound. When you're in front, it's lightning, you hear sound. That's why it's transformation heat, laser absorbed, heat some volume, a very fast thermal expansion produce sound. That's why instead detection, imaging, we can hear cells. How it works? This is blood, this is clots, different origins. It should be rich with red blood cells, it's red, or platelets rich, or mixed, different composition. How it works? How we found mistakenly we can detect it. What's going on? Laser irradiate blood vessels because its absorption red. That's why a lot of light is absorbed by blood. If we create some background, absorption background, it's light, absorption spectroscopy, spectrophotometry. If we have strong background. In our first application to detect very cancer cells, it's problem. We need to reduce this background to detect very rare cancer cells circulating. Circulated tumor cells, it's hot topic right now. Several conferences per month. Because if we can detect, it's another application, we can predict metastasis potentially. Right now, conference San Diego and NIH every single month. But no technology how to do it, different technologies. And how it works? Laser radiate pointer or diode laser, which used in CD players, very small, it's enough for this technology. Irradiate blood vessels, produce signals, transformation in heat and acoustic, which are detected with acoustic transducer, some component of ultrasound technology. That's why it's very simple, it's portable, just laser, irradiate vessels, you produce acoustic signals. We can monitor continuously blood. What we found, usually if cancer cells, bacteria, virus, they produce, we call it positive signals, signals above some background. But occasionally during monitoring few years, what we found? We found some negative signals. We thought it's artifacts, it's noise, it's not related. But after many years collecting data, you focus on research, what we found? It's related with clots. Because when clots inside laser beam, no absorption, no any signals. That's why instead background signals, we have nothing. We call it dark spot or dark contrast or dark mode. That's why it's very easy, it's very simple. If we focused at the beginning on nanotechnology, use gold nanoparticles to target some markers and the platelets-related markers. We're doing right now, but we found that it's completely labor-free. The question is, what's diagnostic, how valuable? Could we develop prototype? Could we bring it market? And could we prevent disease? Or at least to reduce the risk of disease, at least at the beginning. This is typical signal. When a lot of cells together, red blood cells, erythrocytes, they produce positive signals. We can detect aggregation. If we put some drug, we see disappearance. We can detect drug screening, drug efficiency screening very fast. It's real time. When you see negative signals, amplitude gives information how big clots circulating across laser beam. When mixed, it's composition, red or white clotting. Just a few examples. We did an experiment on some genetically modified animal models, several models, infarction, heart attack, sickle, etc. We are working with NIH on these problems. They provide a lot of new models. This is typical. This is background signal from blood. You know, scattering light, absorption, it doesn't matter. There's a strong background. All our 5 years we tried to reduce this blood. If we found vice versa, we need to increase background to increase our sensitivity to detect very small clotting. This is typical signals. When we monitor, this is an infarction model. That's why we see immediately, even before what we found, if before many models, it appeared. That's why what we found. First of all, it's natural physiological process. Aggregation red blood cells, clotting, it depends on viscosity, food, etc. But the question is, we need to detect clots very dangerous above subbackground compared to natural physiological processes. What we are doing right now, it's just a question of statistics. This is typical signal. Usually it takes 1 hour just to detect one clots, dangerous clots, as we said, or some risky. Or in animal models, it's very frequently here. And here you see different patterns. That's why we can identify, because information, it's red rich, or white rich, or mixed rich, give information about origin of diseases, where it happened. Of course, I'm talking about just one application, stroke. It's related with some heart attack, thrombosis, with a nose problem, lung problem, etc. But it's more dangerous. That's my idea. It's very simple where we are. This is experimental prototype, several lasers. It's not portable. Several lasers which irradiate blood vessels in different animals models with different wavelengths, just select signals. And this is our clinical prototype. This is our animal models. We develop technology to identify very easily, to navigate our laser on superficial vessels. Ultrasound, infrared technique, also relatively new. We worked with Milton Weiner. If you remember, he moved to New York, but we developed first prototype. This is our prototype, watch light. This is, it's not clear right now, but contrast, small transducer ultrasound, just few millimeters diameter, foot. This is, we right now using diode laser, a few millimeters diameter, built in this device. But here we use fiber to bring laser from not portable stationary system to irradiate vessels. Sometimes, if it's not so fat, you can see these vessels. That's why navigation is very simple. If we use linear beams to exclude any artifacts, we don't need exact navigation because we need just to cross using laser beams to cross blood vessels. This is how it works. This is our clinical prototype. We developed 2 types. In now we final stage to develop completely portable device. This is just previous model. Where we are. Some general information about our approach. We have many, it's a biomedical engineering laboratory, laser, nanomedicine. I have a list laboratory, not so it's unusual for UAMS. It's engineer, physics, several, not only physician, some engineer, computer engineer who developed this technology. Right now, there is, it's a new diagnostic device. There is no prototype. Many applications, infections, metastasis, and stroke warning. The question is how available is this device. We cannot say right now. We see we can detect, we see links, and nobody, no any statistics right now to predict. If we detect these clots, when it happened, tomorrow, next few minutes, or few years. That's why it's a clinical research device who give us information about could we prevent or could not. Of course, optimistically, we plan to do it, but not in our statistics. No any similar device. There is excellent MRI, X-ray, etc. But this system can detect not moving clots. When it happened, or it's growing, it's also available, but we can detect it fast, circulated clots. The velocity 1 meters per second in large vessels. That's why I know any technique to detect such fast moving object in blood. Especially different vessels. We focus superficial 2-3 millimeters vessels, but ultrasound provide detection in neck area, 1 centimeter diameter. The question is, could we detect small clots, what size, what we are working right now. We have technology, or clinics have pulse Doppler ultrasound. Sometimes it helps. But if familiar with ultrasound, a lot of artifacts, noise, a possibility to detect only large clots. But it works. That's why we can compare. Our sensitivity at animal models, it's approximately few clots in whole volume, 2 milliliter blood or 20 milliliter blood. Its size at diameter vessels 1 millimeter, approximately 20 microns. No any technology can detect it in vivo, even in vitro. Because, if you put blood, it's natural clotting. You cannot say exactly it happened during processing, preparation, or before. This is main problem. We're working with Harvard and Stanford. They developed excellent ex vivo device, but even we discuss its valuable or artifacts during preparation of sample. Cost. Ultrasound device stationary, it's least range. It's 50,000 small one or 150,000. Our device currently costs prototype 60,000, but we didn't plan to use clinical device. It's just experimental device. If you buy diode laser, what we did, it's approximately $25,000 per device. But this is universal system. That's why you can use it for different patients, and cost for one analysis a few dollars right now. We have experience with manufacturing, developing. It's just several companies, my previous device. It's a laser combination with electrostimulation to treat some wound infections or muscle damage, etc. That's why what we have, just 2RB protocol approved. We are working to another one to start to develop clinical prototypes, then clinical trials in UAMS 2012. It's very optimistic prognosis. It's investigational device. We are not going to get FDA approval at this time, but we are working on this. In 2003, because it's very new, we are going to use for detection clotting. The advantage, we don't need any labels. It's completely non-invasive. This is laser. It's completely not harmful for any cells. This is exactly what I told you about technology, about application, about platform. And we found that using this simple, robust device, we can monitor, provide examination, not only blood in lymphatics. There is no any lymphatic diagnostics, because problem with sampling. In vivo, it's much easier. We demonstrate it. It's not for clotting, but it's for basic research to determine the origin of clots and pathway different cells related with these diseases through blood and lymph network. Our stage right now, we applying grant, and we are waiting few $1 million to develop 3-5 prototypes for different applications in focus cancer, simultaneously on clotting. What we found, 50% cancer-related cardiovascular diseases. That's why we'd like to combine it to determine simultaneously circulating tumor cells and clots. Probably that's it. We have several publications when we suggested this technology, and collect statistics to publish our preclinical data on preclinical models. If we're proud, we get several awards. It's first example, use lymph for early disease diagnosis. This is first example application our device for in vivo detection circulating tumor cells. This is some technical issue. This is new nanoparticles developed with Fayetteville, with Cancer Nanoscience Institute. Application for enrichment cells in vivo. We use nanoparticle, inject magnetic nanoparticles, targeted some bad cells. We put magnet, accumulated, clean all cells which cross volume close to magnet. We accumulated tumor cells, we're cleaning infections right now, we are thinking about clotting. Can we collect and move out? We developed several optical methods to improve sensitivity and specificity. Usually our funding it goes from NIH. We have currently 3 R1 grants, Science Foundation grant, E3, we mean 3OD grant. Of course support from Cancer Institute. And lots of collaboration across campus, country. We collaborate with universities in 6 countries. This is our laboratory, main staff. But because broad application, theoretically, we don't have such manpowers. We can work together, collaborate with each department UMS. Because diabetes, infections, cardiovascular inflammation, we can detect macrophage, circulating macrophage in other cells, events, disease-associated events. Several companies provide us nanoparticles. We initial stage to establish our company to create several prototypes for different applications. I think that's it. Thank you for your attention. Just a reminder where I'm from, at least me.
Thank you very much. Questions for Dr. Zaroff?
Can you tell me what your intellectual property position is in all of this?
We got several patents. One patent is pending, and we just discussed about establishing a company who will have all rights to develop this technology. But we are open to collaborate because 2 different applications, general platforms the same, but a lots of modification. Different laser, green, red, infrared, several transducers, several diseases. Of course, we cannot at the same time to cover these medical applications. That's why we will be happy to collaborate. First of all, to develop prototype, general, a second, different medical application. Cancer, infections, and cardiovascular diseases.
Other questions? Dr. Clevenger. While the microphone's going back, I'm the total optimist when it comes to things like this. Is there a chance to sort of take a lot of miniaturization engineering and drive what you have as a prototype into something that is portable, that would work either wirelessly or Bluetooth reporting to your phone that the patient could wear that's a high-risk heart patient, for example?
For application for detection circulated tumor cells for cancer metastasis prognosis, we hope for early screening during mammogram. We analyze women to find some presence of cancer cells in blood is probably much earlier than X-ray or another system determine if we lack with that. Our semi-portable. What does it mean? We have portable system on hand and other parts, ear, like clips, and right now a little large laser, which portable but a little big. You have only on experimental type small prototype which combines small laser, ultrasound, etc. and remote control, detection, analysis. It's huge information like flow cytometry. It's a very simple device. The question is we can monitor continuously this information to identify some very typical signals. That's why if you do even for one patient during a few days, emergency room after heart attack, just to prevent second events or see how drug it works, it's huge information. That's why it's combination very simple technical platform and probably some complicated software to analyze all events, statistics.
So help me think about what you would do with it. What's the clinical significance of the data? So let's say, for example, you started to identify pre-clots, formation of somebody's becoming more susceptible to clots. I guess that's the way to put it. I guess the treatment then would put them on Plavix or blood thinners, right?
Yes.
So, in order to make the decision to use your device, wouldn't you have to already have a preclinical or have some sort of a predisposition? I mean, I don't believe that our society is going to do general screening for something like this. So it'd have to be a predisposed population. And my experience is they get put on Plavix almost immediately anyway. So I'm trying to see where this fits into the clinical relevance.
Thank you for this key question, especially when we expect we have technology which it's very difficult to verify at level sensitivity, at higher sensitivity. Our vision and 3 applications. First of all, it's clear, we found it recently when I visited several counties in California, Becton Dickinson. It's research tool with different animals to study drug screen, to response, clotting, removal, etc. We are working on this. It's nearest. It's clear. It's done. Just to combine together one box and provide many companies ask about it. Second, what I think it's a research clinical device which we provide to emergency room to see what's happening clotting after events, heart attack and stroke, to get clinically relevant significant statistics between clotting and research, stroke. This is just accumulate data to provide. Nobody knows. Maybe we prevent, maybe it's what's prognosis, how long, what's time scale of prognosis. Nobody knows. But definitely it's very helpful to see if we take aspirin, what we see immediately decrease of number clots. That's why it's available device emergency room. Of course, people know to take aspirin, or it's another one. But if it's expensive drug, nobody knows. It's time to start to do it with side effect or no. If finally, I'm not sure it's 3-5 years probably, to develop completely portable device for clinical application first, for risk group, high viscosity, pressure, if some symptoms natural. That's why I provide at least for people with risk of strokes to provide this device to see what's going on with clotting, to prevent last terrible events, at least, to save people. If they start taking therapeutics, probably they exclude this problem, but nobody knows. That's why it's device to recommendation to take immediately strong drug to reduce viscosity, heart blood pressure, etc. That's why our vision, but it's not sometimes quite clear at this stage.
Okay, I think we have to move on. Thank you very much, Dr. Zaro.
Thank you.